With that, I'd like to welcome Dr. Paul Pratt with the U.S. Department of Agriculture Agricultural Research Service to give us the download on Scotch Broom Gall Mite and a perspective from California. So take it away, Paul, and Sarah, we'll ask you to turn your camera off.
Oh, of course.
Hey, this is Paul, and I'm just waiting for my presentation to load here. It may take just a few seconds. But yeah, getting to the point here, my name is Paul Pratt. I work for the USDA, and I'm a product of California State University, Chico, but don't let that make you think less of me, because I am also a product of the Oregon State University Department of Entomology back when that was a thing. I'm currently working for the USDA. My first job after finishing up my master's and PhD at Oregon was a position down in Fort Lauderdale, Florida, where I studied biological control of invasive weeds in the Everglades. And more recently, in 2015, I was invited to return to California, which I jumped at. And now I'm the research leader of the Invasive Species and Pollinator Health Research Unit, where collectively we're 9 scientists. We focus on invasive weed ecology, developing management protocols, including biological control. We also have a scientist that works on pest insects and biocontrol of pests, and then we have 2 honeybee scientists as well. My specific research interests are developing biological control programs, and some of the biocontrol projects that we're working on now include a long list, which I hope you can see here now, ranging from Cape Ivy and Scotch and French broom all the way down to Medusa head, which is a very new project for us, perennial pepperweed, etc. But I want to highlight just quickly that yellow star thistle has been one of the focuses of this lab long before I arrived, but I'm happy to announce that we have released, as of April of 2020, a new root weevil, Seraptapian. Some of you may have heard of that and may be interested in learning more about it, but that's a talk for another day. So let's see, moving on to my next slide. Scotch broom really doesn't need any introduction. We've covered just about everything, including a couple of maps about its distribution in the United States. I just want to reiterate that it is on the East Coast as well, and throughout various other countries, including Australia, New Zealand, South America, Chile, and Argentina, etc. But I wanted to share with you the distribution in California, which kind of plays into the rest of my talk. And that is, this map shows on the right, it shows all those little dots where folks have collected or reported sightings of Scotch broom. And this doesn't necessarily represent the exhaustive distribution of the plant, but it does fairly well represent where the main clusters, if you will, the main populations of this plant are located, kind of in the Sierra foothills, close to the Oregon border, along the coast, down through the Bay Area. So the main character of my talk today is a small areophyid mite called Asteria genisti. And Asteria genisti is a native of Europe. It produces these puffed ball-like galls, and they develop from foliar buds. So these mites are extremely small, and they disperse in the wind. So some have called them aerial plankton or dust in the wind, but they seem to cause quite a bit of damage in their native range. So scientists in New Zealand and then later Australia wanted to investigate the host range of these mites as possible candidates for biological control of the weed in New Zealand and Australia. So back in the early 2000s and late 1990s, they began host range testing this mite and determined that it would not attack any of their native or closely related species in New Zealand and Australia. So in 2007 and then in Australia in 2008, this mite was introduced. It established quickly down under, and fairly quickly there were hundreds of galls per plant, and they observed stem dieback. However, we haven't heard much about the mite since then down in New Zealand or Australia. But curiously enough, this mite was discovered in both Tacoma and Portland in 2005. And that's before I got here in California, so I'm kind of leaning on the the evidence and the hard work of other people that have come before me, and I recognize them at the end of my talk. But there were general predictions that were made. This mite disperses on the wind, so it was assumed that it would diffuse both north and south on those wind currents. And so scientists in California began opportunistically surveying Scotch broom populations along the Oregon border, assuming, I think rightfully so, that that's where it would first show up. But in 2014, the first discovery of the Scotch Broom Gall Mite was found in Georgetown, in the city of Georgetown in El Dorado County, which is in this star. If you can kind of see it here, it's this big star in California that generally identifies the location. Again, my point here, it jumped a huge distance from where the people were surveying along the border all the way down to near Tahoe. So when I arrived in 2015, Mike Pitcairn and Jennifer Andreas and others contacted me and wanted to bring me up to speed on this situation. And so we proposed collectively to address several questions. One was, where is the mite in California? Where is the mite likely to spread? When do the mites disperse from the galls? Of course, they spend most of their time inside these galls, but they have to leave at some point to be blown through the wind and to hopefully land on a susceptible plant, which they can then gall. So do the mites affect plant performance? And, very importantly, are the mites host specific? And unfortunately, I don't have time to give you all of the data and graphs for each one of these questions. I'm going to kind of cut things a little bit short, and I'm happy to share additional information on a sidebar. But I'll go through each one of those questions and provide kind of an update, if you will. So we conducted a survey, myself, some folks in my lab, Mike Pitcairn and other individuals in the state, and visited as many Scotch broom populations as we could from 2015 through 2017. But we also employed this online reporting system where folks could email content and information, including photos, to a website, and then we would gather that information, and collectively we were able to develop the map on the right, which includes populations observed along the northern part of the state, a couple of scattered populations, around Mount Shasta and Redding, and then over here in Mendocino, we found 3 plants that had a few galls each. But the vast majority of the galled plants that we observed, which is really the hallmark of this mite, is these large galls developed on the vegetation. This was primarily found in the Sierra Nevada foothills, which led us to assume that these large populations, almost continuous populations across 7 different counties in that belt of Scotch broom, represents an early invasion, likely the very first invasion in California. These other invasions or these other populations that we observed are much more likely to be dispersing at a later date and have yet to expand into large populations. That could be wrong. It could be affected by temperature. It could be affected by nutrition of the plants, but that's our working hypothesis at this time, suggesting that maybe this population in the Sierra Nevada foothills was actually transported on machinery, equipment, lumber, materials, things like that, and accidentally introduced into that zone. So the next topic we wanted to address was predicting the distribution, and we did this by using the software MaxEnt. Some of you may have heard of it. It's free software. You input data on the presence, where the mite is for sure, where it's established. And then it uses an algorithm based on elevation and climate and things like that to determine where else it might go. So for the presence data, we gathered information from the US, where we had knowledge of its distribution, also Australia and New Zealand, where it was introduced as a biocontrol agent, and then its native range. So we had a total of 135 GPS points documenting the known distribution of the mite, and then we modeled that using the software. And climatically compatible areas of Scotch broom sites were then identified in this map on the right. And I'm just going to draw your attention to the gray area. That's kind of where Scotch broom and the mite are predicted based on the model to overlap. And I'm happy to report in Washington, that's most of the invaded area seems to be climatically suitable for the mite. In Oregon as well, California, you'll see some incompatibilities there where it's red or it's blue. Blue, if you can't read the fine print, and I apologize for that, blue represents areas where the plant is predicted based on the model to persist or grow well, but the mite isn't climatically suitable or won't persist there as well. So the next question we wanted to address, jumping to the next topic, is when do the mites disperse? When do they leave these galls? You can see in the bottom picture, that's a SEM, a cross section of a gall. I just cut it in half and put it into the SEM, and you can see all of those areophyid mites that kind of look like worms, and then the spherical objects are their eggs. There are just hundreds of thousands of these individuals of all stages inside these galls. And so what we did is we just sampled this material monthly on a regular basis over 2 years, and then washed the samples. Then we rinsed those samples through a series of sieves, and then we filtered that material and then looked at the filtered material under a microscope so we could count the number of mites. And I'm sorry to say, well, I'm sorry to say for my technicians who were processing these samples, month after month after month, they were usually 0. However, we did see a very large spike in the number of mites over only a 2-month window. It was really surprising. So this kind of shows, if you will, a cartoon example of when the plant is producing new growing leaves, which would be susceptible to mite. Remember that this might feed on new buds. Those new buds then start expanding, and the mite causes the leaves and the stems to be deformed into this gall. So there needs to be actively growing buds for the mite to exploit. And so what we saw was there was this long period of dormancy, if you will, during the middle of the summer. However, we did see a dramatic pulse of mites dispersing, and you would assume that they would be phenologically associated with this new growth of Scotch broom, but we saw just the opposite, that the mites seemed to be coming out over a 2-year period. They came out consistently during the middle of the summer. They peaked in July. We saw some in June, but most of them in July and August, which is months before the plants became susceptible, in my view. So it's curious, what is going on? Why are these mites dispersing so early and prematurely? So the next question that we had, does galling influence plant performance? And so I set up 2 experiments. One was, hey, we can use a miticide. We can kill the mites on some plants. On a plant right next to it, we won't treat it at all, and we'll just compare plant biomass between their different fragments. So we could look at stems and leaves and galls and fruit on plants that have been treated or protected from the mite, and those that have had mites, populations allowed to feed and attack and change their biomass theoretically. But we've collected those data. We did that over a 2-year period. We collected those data, and I haven't had a chance to analyze the data, but I want to unfortunately report that looks like I lost my presentation there. So I'm sorry to say that it doesn't appear that the mites had much of an effect, at least without running the statistics. Just visually, I can tell there's not a real difference in that 2-year study. However, at the same time, at 3 different sites in the Sierra Nevada foothills, we also did these long transects and harvested 50 plants at each site, so a total of 150 plants, ranging in size from small seedlings all the way up to these very large individuals taller than myself. And we categorized them into heavily galled plants or plants that had very light or no galls, so 1 or 2 galls or 0 galls. And we compared the biomass of those same fractions. And what we saw was no difference actually in stems or branches or leaves. But we did see a significant difference in fruit biomass. So plants that had heavy galling produced markedly fewer fruit than those that didn't. All right, the last question I'm going to address, and hopefully I'm doing okay on time. The last question I'm going to address is that is this environmentally safe? This new mite that's arrived in Oregon first, in Oregon and Washington first, and then has recently dispersed into California. Is it safe? Is it going to attack native lupins? Is it a beneficial organism because it'll attack not only Scotch broom, but maybe some of the other brooms? So what better way to do this than to go out into the field where there are already galled plants and look in the vicinity of these plants to determine if there are any other plants getting galled, including lupins or other close relatives, as well as the other brooms, which often in California occur sympatrically. So these, the map here where the stars are, you can almost see those maybe in the screen. Those represent the locations that we surveyed, looking at the galled plants and then any natives in the vicinity. And I'm sorry to report that we didn't see any galls on any of the brooms. So we looked at Portuguese, Spanish, and French broom, in addition to Scotch broom, never saw a single gall on any of those other exotic weeds. But we also didn't see any galls forming on any of the lupins, which was encouraging and good. So
3 minutes,
because
3 minutes.
Okay, I think I'm going to make it. So we also started 2 other experiments. They're common garden tests, if you will. We took 12 perennial species and we just dumped thousands of mites onto 6 replicates of each of these perennial species. And then we monitored those plants for 12 years. We did the same thing with 10 annual species and released mites directly onto the plants. They didn't have to disperse in nature. We did the dispersing for them. And we saw no gall development on any of the lupins, the perennials or the annuals, but we did observe that the mites were able to live and persist on the plants for several weeks, up to 6 weeks on some of the annuals, which I found quite surprising. So I'm going to review my conclusions here, and one of them is that the mite is very well established in California. I understand, but I haven't seen it personally in Oregon and Washington, that it's very well established there as well. But the model suggests, at least, and if you can believe models, the model suggests that there's great potential for additional spread. And so as I hear about Scotch broom surveys and assessments, I'd be interested in seeing how we can marry these 2 ideas of surveying for the Scotch Broom Gall Mite, see if we can piggyback that on to some of the work that's already being done, just to better understand the geographic distribution of this mite. I want to announce that the East Coast doesn't seem to be a good climate match based on the model, but South America does. So both Chile and Argentina showed up as potential environmentally suitable habitats for this mite. So the mite disperses long before the plant becomes susceptible, which makes it available or exposed to predators. And this issue of phenological synchrony has me wondering about resistance or selection. And I'm really interested to hear the talk later on today about that exact same topic. So chronic galling clearly reduced reproduction. Plants appeared to compensate when their foliage was damaged, but they compensated at the cost of reproduction. And we did see anecdotally in the field that the gall mite killed stems. We saw entire plants killed in certain instances, but it's impossible to know if this is due exclusively to the mite or some various other environmental factors that might be going on. So experimentally, I have not been able to demonstrate that to date. All right, no galls were produced on any of the other brooms, unfortunately, but no galls in the field were found on lupins. Prolonged survival of the gall mite was observed on some annual lupin leaves, but we never saw any visible damage. However, I need to note that others have observed abnormal growth, specifically on Lupinus florus in a greenhouse experiment. We've tried this twice. We haven't been able to replicate that yet, but it may be host plant material. So we need to get that plant material that was used to test to make sure that we're talking about the same species or the same biotype and see if we can replicate that. But I want to conclude by saying that evidence suggests that Aceriogenicity is very host specific and is likely to be safe for redistribution. But don't do it, because it requires a permit. It needs a federal permit to redistribute the mite. So one of my questions remains, that permit requires a lot of time. You have to put in a lot of energy. It can take 2 or 3 years for us to get a permit to move the mite from one part of Washington to another. So will the mite spread fast enough to justify going through the permitting process? And it would be helpful to get additional data from those individuals who are listening today as to its ultimate distribution and the value of that permitting process. All right, I just want to acknowledge a whole suite of people. Jenna Andreas, of course, is on here. Mike Pickern and Scott Oneto from CDFA and the extension program here in California. And then my colleagues in Australia and New Zealand as well were super helpful in getting all of this data collected and gathered. And finally, I want to list my email address because I welcome input and insight from others that might have additional information. As I said, I'm kind of new coming back to California and not nearly as familiar with Scotch Broom as those individuals that are participating in this meeting today. So I look forward to hearing your input. Thanks.